Welcome back, class. This is Silvana Iconi with Lecture 5, the viruses. So in this lecture we're going to focus on the broader aspects of virology, that is the study of viruses. We'll look at a brief history of the discovery of viruses, their general properties and the structure of viruses, the ways in which they're cultured, and very briefly, viral taxonomy. We'll move on to a new topic now, the cultivation of viruses. So because viruses are unable to reproduce independently of living cells, they can't be cultured in the same way as prokaryotic and eukaryotic microorganisms. So we can't just make up nutrient media and culture the viruses in petri dishes. Cultivating viruses requires inoculation of appropriate living hosts. For many years, researchers have cultivated animal viruses by inoculating suitable host animals or embryonated eggs, that is, fertilised chicken eggs incubated about 6 to 8 days after laying. And that's what's shown here in this image. So to prepare the egg for cultivation of viruses, the surface of the shell is first disinfected and then penetrated with a small sterile drill. After inoculation, the drill hole is sealed with gelatin and the egg is incubated. Some viruses reproduce only in certain parts of the embryo, so the specific virus has to be injected into the proper region. More recently, animal viruses have been grown in tissue or cell culture on layers of animal cells. And this technique is made possible by the development of growth media for animal cells and by the use of antimicrobial agents that prevent bacterial infection and fungal contamination. The viruses are added to a layer of animal cells in a specially prepared petri dish and allowed time to attach to the cells. The cells are then covered with a thin layer of agar to limit the spread of virions so that only adjacent cells are infected by newly produced virions. And as a result, localized areas of cell destruction and cell lysis called plaques are formed, and these may be detected if stained with dyes, so that they can be distinguished from living cells. Now, viral growth doesn't always result in cell lysis to form a plaque. Animal viruses in particular can cause microscopic or macroscopic changes or abnormalities in host cells and in host tissues. And these are referred to as cytopathic effects, or CPEs. Cytopathic effects may be lethal to the cells or tissues, but plaque formation from cell lysis doesn't always occur. In the photos in A and B to the left of screen, we can see a couple of examples of virus plaques. In A, we can see some clear plaques caused by cell lysis due to poliovirus infection. And the animal cells that are being used here are monkey kidney cells. In B, to the left of screen, we can see the effect of bacteriophage infection in bacterial cells, and the plaques in this case have been made visible by staining with a red dye such as neutral red. In A, B and C to the right of screen, we can see what happens when a particular cell type undergoes cytopathic effects. So the cell type that's being used in this study are fibroblast cells from fetal tonsils. In A, you can see the normal morphology of fibroblast cells. In B, you can see the cytopathic effects caused by infection of these cells with adenovirus. And in C, different cytopathic effects caused by infection of fibroblasts with the herpes simplex virus. Viruses that infect bacteria and archaea are normally cultivated in either nutrient broth or nutrient agar cultures of young, actively growing cells. In some infected cultures, so many of the bacterial or archaeal host cells are destroyed that a normally turbid looking culture very rapidly clears because of cell lysis. Agar cultures are prepared by mixing viruses with cool liquid agar and a suitable culture of host cells. Then the mixture is quickly poured into a petri dish containing a bottom layer of sterile agar, and after hardening, the cells in the layer of top agar grow and reproduce, forming a continuous opaque layer or lawn. Wherever a virion comes to rest in the top agar, the virion infects an adjacent host cell and reproduces. Eventually, lysis of the cells generates a plaque or clearing in the lawn of cells. And of course, as shown in the photos to the right of screen, the appearance of the plaque is often characteristic of the specific virus that's being cultivated. Let's look at hosts for cultivating plant viruses. Plant viruses can be cultivated in a number of different ways. So for example, in plant tissue culture, in cultures of separated cells, or in cultures of protoplasts. Protoplasts are plant cells lacking cell walls. Viruses can also be grown in whole plants. So for example, leaves can be mechanically inoculated with an abrasive such as carborundum, which is a carbon and silicon abrasive. The photo in A shows a leaf of Nicotiana glucinosa, which is infected with the tobacco mosaic virus. So what's the role of the abrasive? The abrasive breaks the cell walls of the plant, and when this occurs, the virus can directly contact the plasma membrane and infect the exposed plant cells. In nature, the role of the abrasive is frequently filled by insects that suck or crush plant leaves and thus transmit viruses in that way. So once the plant cells are infected with the virus, a localized necrotic lesion often develops due to the rapid death of cells in the infected area. Even when lesions don't occur, the infected plant may show symptoms such as change in pigmentation or changes in leaf shape. Some plant viruses can only be transmitted if a diseased part of the plant is grafted onto a healthy plant. The photo in B shows tobacco mosaic virus infection of an orchid, and it shows the characteristic leaf colour changes that occur. Now, virologists must be able to purify viruses and accurately determine their concentration in order to study virus structure, reproduction, and other aspects of viral biology. These methods are so important that the growth of virology as a modern discipline depended on their development. So, virus purification makes use of several properties of viruses. One, virions are very large relative to proteins. They're often more stable than normal cell components, and they have surface proteins. So because of these characteristics, many techniques useful for isolating proteins and cell organelles can also be employed to isolate viruses. 4 of the most commonly used approaches are listed here: differential and density gradient centrifugation, precipitation, denaturation, and enzymatic digestion. So differential and density gradient centrifugation are often used to first separate the virus particles from the infected host cells. In differential centrifugation, cells are first disrupted in buffer, then centrifuged at various speeds to separate particles of different sizes. First, they're centrifuged at 100,000 G to sediment the virus particles and the heavier organelles. Then the supernatant is removed with the smaller particles. Then the suspension is centrifuged again for 10,000 G for 20 minutes to sediment the cell organelles while leaving viruses in solution. Then 100,000 G again for 3 hours to sediment and concentrate the viruses. Density gradient centrifugation is very similar, but it uses a gradient of sucrose from about 20 to 50% sucrose. And then when centrifugation takes place, each virion particle will come to rest at a density which is equal to the density of sucrose. And although these two procedures remove most of the host cell material, some cell components may still remain. Then we can use precipitation using, for example, ammonium sulfate at a concentration just below that needed to precipitate the virus particles. So many of the cell components, that is the host cell components, are precipitated while the virus remains in solution. Viruses tend to be less sensitive to heat or pH than cells are. So solvent treatment like chloroform or butanol can be used to denature any contaminants. The aqueous phase contains the suspended virus, while the organic layer contains any dissolved lipids. Then any remaining cell proteins and nucleic acids can be enzymatically digested using enzymes such as ribonuclease and trypsin. And even though viruses have a protein coat, these proteins are usually more resistant to attack by these enzymes. The number of viruses in a sample can be determined either directly by counting virus particle numbers or indirectly by measurement of an observable effect of the virus. And the values obtained by these two different approaches often don't correlate closely, however they're both of value. The virions can be counted directly using an electron microscope, as shown in this image here. In one type of direct count procedure, the virus containing sample is mixed with a known concentration of small white latex beads, as you can see in this image. And then they're sprayed onto a coated grid. Both the latex beads and the virions are counted, and then the virus concentration can be calculated from these counts and from the bead concentration. And the indirect method of counting virus particles is called the hemagglutination assays. So we know that many viruses can bind to the surface of red blood cells. And if there's enough of them, the red blood cells will clump together and settle out of suspension, and this is termed agglutination. So the red blood cells are mixed with a series of virus dilutions, and then each mixture is examined. The highest dilution that causes agglutination to occur is referred to as the hemagglutination titer. A variety of indirect assays can be used to determine virus numbers in terms of infectivity, and many of these are based on the same techniques used for the cultivation of viruses. So for example, in the plaque assay, several dilutions of viruses are plated out with appropriate host cells, and when the number of viruses plated are much lower than the number of host cells available for infection, and when the viruses are distributed evenly across the plate, then each plaque that arises in a layer of host cells is assumed to have arisen from the reproduction of a single virion. Therefore, a count of the plaques produced at a particular dilution will give the number of infectious virions. And this number is referred to as plaque forming units, or PFU. And the concentration of infectious units in the original sample can then be easily calculated. When biological effects are not readily quantified using these techniques, the amount of virus required to cause disease or death can be determined by what's called the endpoint method. Using this method, organisms or cell cultures are inoculated with serial dilutions of a virus suspension. The results are used to find the endpoint dilution at which 50% of the host cells or host organisms are killed. The lethal dose, known as LD50, is the dilution that contains a dose large enough to destroy 50% of the host cells or organisms. In a similar sense, the infectious dose, or ID50, is the dose that, when given to a number of hosts, causes an infection of 50% of the hosts under the conditions employed. Here we have a hypothetical dose-response curve used to determine LD50, or lethal dose 50. Percentage deaths of host organisms or cells are found on the y-axis, and the virus dilution used is found on the x-axis. The LD50 is indicated by the dashed line, and we can see the dilution of 10 to the minus 6 is the LD50. Let's finish now with a brief look at virus taxonomy. The classification of viruses is difficult, and it's far less advanced than that of cellular microorganisms. In part, this is due to a lack of knowledge of their origin and evolutionary history. So in 1971, the International Committee for Taxonomy of Viruses, or ICTV, developed a uniform classification system. Since then, the number of viruses and taxonomic categories has continued to expand. In its 8th report in 2008, the ICTV described almost 2,000 virus species and placed them in 3 orders, 73 families, 9 subfamilies, and 287 genera. The committee placed greatest weight on specific properties to define families. For example, nucleic acid type, nucleic acid strandedness, the sense of single-stranded RNA genomes, that is positive strand or negative strand, the presence or absence of an envelope, the symmetry of the capsid, and the dimensions of the virion and capsid. This table summarises some of the common virus families and the characteristics used to classify them. Let's look at just one example. The first one in the table, Picornaviridae. Genome size is 7 to 8 kilobases, nucleic acid type RNA, strandedness, the RNA is single-stranded, capsid symmetry, icosahedral, number of capsids 32, presence of an envelope, Picornaviridae lack envelopes, size of capsid is noted, and the host range A tells us that the Picornaviridae infect animals. Let's finish with a look at an example of viral nomenclature. Virus names are derived from various aspects of their biology and history, including maybe features of their structure or diseases they cause, and maybe even locations where they were first identified or recognised. Virus order names always end in virales. So if we look at the example here at the top of the screen, order mononegavirales, meaning single and negative. And this refers to a group of related viruses containing negative strand, single-stranded RNA genomes. This order contains 4 families, including the one shown here, family Paramyxoviridae. So virus family names always end in viridae. Paramyxoviridae means by the side of, and mucus. This family contains 2 subfamilies, and virus subfamily names always end in virinae. Paramyxoviridae, which contains 6 genera, including the 2 shown here, genus Rubulivirus and genus Morbillivirus. Genus and species names always end in virus. One example in the genus Rubulivirus is the species referred to as type species mumps virus. And an example of a type species belonging to genus Morbillivirus is the measles virus.